So what if the microscopic delivery vans that are designed to save immunocompromised patients are actually carrying the exact molecular triggers that cause their bodies to reject the treatment?
Yeah, it's a terrifying thought,
It
honestly.
really is. I mean, we hear constant hype about exosomes and nanocarriers.
Yeah.
Everyone says they're the ultimate future of targeted drug delivery and regenerative medicine.
Right, because the promise is that we can heal tissues and deliver payloads without all the clunky, dangerous side effects of whole cell therapies.
Exactly. But today, we're asking a question that is basically keeping immunologists and biotech developers up at night. Could injecting these biological messengers into highly vulnerable hosts actually trigger, like, systemic HLA sensitization and alloimmune reactions?
And that right there is the critical hurdle for the entire field of nanomedicine right now, because when we operate at the cellular and subcellular level, the rules of immune tolerance get incredibly complex.
Yeah.
We are looking at data showing that what we previously thought of as just inert biological debris actually has the power to fundamentally reprogram a recipient's immune system.
So we'll get to the bottom of this. We are looking at a stack of research today covering extracellular vesicles in organ transplantation, the immunology of mesenchymal stromal cell therapies, MSCs, and some highly counterintuitive data on how the immune system actively hunts down completely synthetic nanocarriers.
Oh, that synthetic data is fascinating.
It's wild. Our mission here is to unpack the exact mechanisms of exosome-host interactions, discover the hidden triggers of alloimmunity, and really determine if these breakthrough therapies are inadvertently acting as Trojan horses.
Which is a huge deal for anyone in this space.
Exactly. So whether you're actively developing these therapeutics or you just love tracking the cutting edge of clinical immunology, this deep dive is going to completely reframe how you look at drug delivery. So let's start with the baseline mechanics. When an allogeneic exosome enters a recipient's bloodstream, how is the immune system actually processing it?
Well, to understand that, we have to look at the three distinct pathways of allorecognition. Exosomes are essentially lipid-bound extracellular vesicles packed with RNA, proteins, and metabolites.
Basically just tiny biological bubbles.
Yeah, exactly.
Yeah.
Tiny bubbles. But crucially, they carry the human leukocyte antigens, or HLAs, and major histocompatibility complexes, MHCs, of their source cell. So when they enter the host, the first pathway is direct allorecognition.
Okay, direct.
Right, and this just involves the intact donor exosome directly cross-linking with the recipient's T-cell receptors.
Got it. The direct physical interaction. That seems straightforward enough.
It is. The second is the indirect pathway. This is where the recipient's antigen-presenting cells, like dendritic cells or macrophages, internalize the foreign exosome, process it internally, and then present the resulting peptide fragments on their own endogenous MHC molecules to activate the T cells.
So that's just standard antigen processing. The cell eats the garbage and shows it to the immune system.
Precisely. But the third pathway is where the architecture of the immune response gets really strange. The literature refers to it as the cross-dressing phenomenon.
Cross-dressing.
Yeah, or the semi-direct pathway. In this scenario, the recipient's antigen-presenting cells capture the donor exosomes, but they don't break them down.
Wait, they don't eat them.
No. Instead, they physically fuse with the vesicle and insert the donor's intact MHC molecules directly into their own lipid bilayer.
Oh, wow. So the recipient's immune cells are basically putting on the donor's name tag and walking around the body, tricking the T cells into sounding the alarm.
That is a perfect analogy, yes. The host's own cells become biological imposters. They're circulating through the lymphatic system, displaying a foreign immunological identifier, actively tricking the host's T cells into initiating a systemic alloimmune cascade against those specific donor markers.
But wait, I have a problem with this mechanism. Because exosomes are denucleated vesicles. Like we said, they're just lipid bubbles.
Right.
How does an isolated vesicle cross-link T-cell receptors effectively enough to initiate a full cascade without the requisite co-stimulatory machinery, like CD80 or CD86 that you would normally find on a live antigen-presenting cell?
Yeah.
I mean, how does cellular debris start a war?
It's a really critical distinction, and honestly, it was a point of heavy debate in the literature for a while. The studies clarify this by comparing in vitro and in vivo models.
Okay.
Because in an in vitro assay, Isolated allogeneic exosomes do bind to T cells, but they completely fail to stimulate proliferation. They lack that secondary co-stimulatory signal you mentioned. They essentially knock on the door, but the T cell doesn't fully activate. They absolutely require the presence of the recipient's antigen-presenting cells to complete the circuit.
So in a vacuum, or like in a controlled lab dish,
Sure.
the exosomes alone are insufficient.
Exactly. But you move that into an in vivo model, a living, breathing host, and the dynamics completely shift, and it hinges entirely on the baseline inflammatory state of the recipient.
Context matters.
Massively. The research demonstrates that allogeneic exosomes actively trigger T-cell proliferation and sensitize hosts to alloantigens only when they are delivered into an inflammatory microenvironment.
Okay, and this is the crux of the issue for clinical application, isn't it? Because if you are developing an exosome therapeutic, you're not usually targeting healthy individuals with pristine immune systems. You are targeting patients with active tissue damage, organ transplants, severe autoimmune flare-ups.
Right, you're treating sick people.
Yeah, so you're deploying these vesicles into an environment saturated with damage-associated molecular patterns, or DAMPs.
And DAMPs completely alter the threshold for immune activation. They are basically the biochemical distress signals released by stressed or necrotic tissue. So when you introduce donor exosomes into a space flooded with DAMPs, the host's antigen-presenting cells are already hyperactivated. They readily snatch up those exosomes, engage in that cross-dressing pathway we talked about, and present the donor M8Cs to a T-cell population that is already primed for aggression by the systemic inflammation.
It's basically like throwing a match into a powder keg.
Yeah.
And there is a clinical case study in our sources that perfectly illustrates how catastrophic this synergy between inflammation and exosome presentation can be.
Oh, the lung transplant patient.
Yes, the 76-year-old female patient who had received a lung transplant. She was on a heavy regimen of immunosuppressants to prevent acute rejection. And then she contracted SARS-CoV-2.
Yeah, this case is so telling. During her viral infection, researchers isolated and analyzed her circulating exosomes, and they found that these vesicles were heavily loaded with SARS-CoV-2 spike proteins.
Which makes sense, right? That is a standard physiological response. Her cells were processing the viral threat.
Exactly. But the crucial finding, the real twist here, was that these same exosomes were concurrently carrying lung self-antigens, proteins specific to her transplanted graft, alongside high levels of HLA-DR and DQ molecules.
Oh, wow. So the viral infection turned her own exosomes into this highly immunogenic broadcast system. They were simultaneously displaying the viral threat and the tissue markers of the transplanted lung.
Yeah, and the clinical outcome is what makes this such a cautionary tale. Because she actually cleared the COVID-19 infection, her symptoms resolved, and the viral spike proteins eventually disappeared from her circulating exosomes.
But she didn't get better.
No. The exosomes carrying the lung self-antigens and HLA molecules persisted at elevated levels. Her immune system had established a new, highly aggressive memory against the allograft. Her lung function steadily declined, which led to chronic lung allograft dysfunction, or CLAD.
That is terrifying, but so fascinating. The inflammation from the virus was the catalyst, but the exosomes served as the biological memory of that immunological trauma.
Yes.
So long after the DAMPs from the virus were gone, the cross-dressed antigen-presenting cells continued to drive alloimmunity. This proves that we really cannot look at the exosome therapeutic in isolation.
Not at all.
I mean, the exact same vesicle that might be completely tolerated by a stable patient could trigger irreversible graft rejection if that patient happens to be fighting off a mild pathogen at the time of injection.
Which is exactly why the field is increasingly focused on the microenvironment. The baseline immunological context basically dictates whether an exosome is interpreted as a therapeutic signal or an invading pathogen.
Well, given the severe immunogenicity of biological exosomes in inflammatory microenvironments, I have a logical follow-up. Why not just pivot to synthetic carriers?
Right.
I mean, if donor HLA molecules are the primary trigger for sensitization, then moving to completely synthetic empty nanocarriers like pegylated liposomes should theoretically bypass the alloimmune reaction entirely. You remove the protein, you remove the problem.
Right,
Yeah.
you would think so. And honestly, that was the prevailing assumption for a very long time. But the pharmacological data on synthetic nanocarriers presents a different, equally formidable challenge. It's called the accelerated blood clearance, or ABC phenomenon.
Okay, what is that?
Well, it turns out that even devoid of biological proteins, synthetic carriers are highly immunogenic.
Like, really? But the industry standard for these synthetic carriers relies heavily on pegylation. Polyethylene glycol is layered onto the surface of the lipid nanoparticle to act as a steric shield.
Mm.
It's literally supposed to hide the carrier from the immune system and prevent opsonization so the drug can actually reach its target.
And that stealth coating works perfectly for the initial administration.
Oh, just the first one.
Right. But cross-administration studies involving repeated injections revealed a massive flaw. That first stealth dose actually induces the splenic marginal zone B cells to produce anti-PEG IgM antibodies.
Wait, so the immune system isn't ignoring the invisibility cloak. It is actively synthesizing a targeted weapon against the chemical structure of the cloak itself.
Exactly.
But why does a synthetic polymer trigger a B cell response? Like we said, there are no proteins to present.
Because the immune system is essentially a highly sophisticated pattern recognition machine. It doesn't solely rely on peptide presentation. The spatial arrangement of the PEG polymers on the surface of a liposome creates this dense repeating chemical array. And to the immune system, this dense polymeric array structurally mimics the T-cell independent type 2, or TI2, antigens that are typically found on the polysaccharide capsules of certain bacteria.
Oh man, so it's a structural false positive. We engineered a synthetic surface that accidentally perfectly mimics the geometry of a pathogen.
Yes. So when a second dose of that pegylated liposome is administered days or weeks later, those circulating anti-PEG IgM antibodies immediately bind to the synthetic surface.
And then what happens?
This binding rapidly activates the classical complement pathway. C3b fragments coat the liposome, which is a process called opsonization, and that signals the Kupffer cells in the liver to aggressively clear the nanocarriers from the bloodstream before they can deliver any payload.
Wow. And there was a fascinating detail in the sources regarding a study on beagle dogs that really highlights how sensitive this mechanism is to physical geometry.
Oh yeah, the hierarchy study.
Yeah, they didn't just test liposomes, they tested a whole hierarchy of synthetic structures to see what drove the strongest ABC response.
And the structural data is incredibly revealing. The strength of the immune clearance from strongest to weakest went from pegylated liposomes at the very top down to emulsions, then solid lipid nanoparticles, and finally PEG micelles, which triggered the weakest ABC response.
But why such a drastic difference if they all utilize PEG? I mean, it's the same coating.
It comes down to rigidity and surface density. Liposomes have a rigid, highly organized lipid bilayer architecture. When PEG is anchored to that rigid structure, it presents that dense repeating array that strongly cross-links the B-cell receptors.
Right, the TI2 mimicry we were talking about.
Exactly. Micelles, on the other hand, are more dynamic. They lack the rigid bilayer, meaning the PEG polymers are less densely packed or structurally fixed, leading to a much weaker TI2 mimicry and consequently far less IgM induction.
So it proves that engineering our way out of sensitization isn't as simple as just stripping away the biology.
Not at all.
Whether the pattern is a donor MHC molecule on a natural exosome or a dense array of synthetic polymers on a liposome, the immune system will learn it and mount a systemic attack upon re-exposure.
The host defense mechanisms are incredibly plastic. You cannot simply bypass them with inert materials if the geometry still triggers evolutionary alarm bells.
Okay, so let me synthesize this for a second because we're painting a rather daunting picture of nanomedicine here. Biological exosomes can trigger severe alloimmunity and HLA sensitization via cross-dressing, and they become highly dangerous in the inflammatory environments typical of the patients who need them most.
Right.
Meanwhile, fake synthetic nanocarriers get neutralized by anti-PEG IgM and the complement cascade. So why on earth are biotech portfolios currently overflowing with exosome startups? Why is so much capital and research pouring into a delivery mechanism that seems inherently prone to triggering immune rejection?
Because of the therapeutic paradox, the immunogenicity of a biological exosome is entirely dependent on its cellular origin.
Okay, explain that.
Well, everything we've discussed regarding HLA sensitization and cross-dressing largely applies to exosomes derived from mature dendritic cells or inflamed tissues. But exosomes derived from mesenchymal stromal cells, MSCs, or regulatory T cells, TREGs, operate on a completely different paradigm. They are perhaps the most potent targeted immunosuppressants we have ever discovered.
So the flip side of the coin.
Mm-hmm.
The delivery architecture is the same, but the internal payload dictates an entirely different systemic reaction.
Exactly. Let's look closely at MSC-derived exosomes. Instead of carrying antigens that prime the immune system for attack, they are loaded with highly specific microRNAs, particularly miR-142-5P and miR-155P, along with a suite of anti-inflammatory metabolites.
Wait, how do those specific microRNAs alter the recipient's immune response at a mechanical level.
It's brilliant, really. When an MSC exosome is internalized by a recipient's antigen-presenting cell, those microRNAs are released into the cytoplasm. They specifically bind to the 3' untranslated regions of the target mRNAs that encode for co-stimulatory molecules, specifically CD80 and CD86.
Okay.
And by binding to those mRNAs, they actually degrade them or inhibit their translation entirely.
Wow, so it's transcriptional silencing. The antigen-presenting cell might still capture foreign debris, but the MSC exosome has essentially severed the wiring.
Yes, exactly.
Without CD80 and CD86 surface proteins, the APC cannot provide that required secondary signal to activate the T cells.
Right, so they induce a state of energy or non-responsiveness in the T cell population, but they go further than simply shutting down activation. These MSC exosomes actively reprogram the cellular environment.
How so?
They induce a phenotypic shift in the recipient's macrophages, driving them away from the aggressive inflammatory M1 state and forcing them into a tissue-healing anti-inflammatory M2 state.
Okay, but if MSC exosomes are this effective at downregulating antigen presentation and shifting macrophage phenotypes, are we just inducing a different kind of risk?
What do you mean?
Well, by systemically administering these suppressive exosomes, aren't we potentially blinding the immune system to opportunistic infections or circulating malignancies?
Ah, I see. That is a primary concern with traditional broad-spectrum immunosuppressants like calcineurin inhibitors. But the data suggests MSC exosomes provide a much more targeted immunomodulation.
Okay, so it's more precise.
Very much so. They don't ablate the whole immune system. They restore homeostasis by specifically inducing apoptosis in the hyperactivated aggressive T cell clones that are driving the immediate inflammation while leaving the resting immune repertoire largely intact.
And we actually see this distinct advantage playing out right now in clinical trials for graft versus host disease, or GVHD.
Oh, definitely.
Because in severely immunocompromised transplant patients, the donor stem cells recognize the host tissue as foreign and begin a systemic attack. Historically, one approach was to infuse the patient with whole mesenchymal stromal cells to try and calm that reaction down. So why is the field pivoting so aggressively toward MSC exosomes instead of using the whole cell?
Well, infusing whole living MSCs into a severely inflamed, immunocompromised patient carries immense physiological risks. Whole cells are massive on a microscopic scale.
Right, compared to a tiny exosome.
Exactly. When infused intravenously, they frequently cause microvascular obstruction, like literally clogging the pulmonary capillaries. Furthermore, because they're living cells, there is a severe risk of ectopic engraftment, where they take root and calcify in the wrong tissues.
To use a computing analogy, deploying whole MSCs is like installing massive, clunky new hardware into a fragile system. It might not fit, it could short-circuit the motherboard, and it introduces entirely new vulnerabilities.
That's a great way to put it.
But utilizing exosomes is effectively sending a targeted software update. You get all the necessary immunomodulatory programming, the microRNAs, the transcriptional silencing of CD80, without the physical hardware risks of circulating living cells.
Because it's a cell-free therapeutic, they easily navigate the microvasculature, they cannot form ectopic tissues because they're not alive, and they deliver their pacifying payload directly to the hyperactive antigen-presenting cells driving the GVHD. The duality of the exosome is just incredible. The exact same biological architecture can either act as the catalyst for devastating HLA sensitization and allograft rejection via cross-dressing, or it can serve as the ultimate immunomodulator, capable of shutting down aggressive autoimmune cascades.
Yep.
And it relies entirely on the precise cellular origin of the vesicle and the underlying inflammatory state of the host receiving it.
It really demands an unprecedented level of precision from clinical medicine. We can no longer treat exosome therapies as a broad category. We have to meticulously map the internal cargo, the surface MHC expression, and the patient's existing DAMP profile to ensure we are triggering targeted tolerance rather than systemic rejection.
This actually brings us to a really provocative frontier. If we fully understand how exosomes transfer immunological memory and how they can transcriptionally silence specific immune pathways, could we engineer a completely new class of therapeutics designed to reverse existing damage?
That is the horizon line for this research. If exosomes are so fundamentally effective at altering how the immune system views what is self and what is foreign, the next logical step is engineering eraser exosomes.
Eraser exosomes. What exactly would an eraser exosome be targeting?
Well, consider a highly sensitized patient, someone who has had multiple blood transfusions or a previous failed transplant. Their bloodstream is flooded with circulating alloantibodies, making it nearly impossible to find a compatible donor match.
Right. Their body is primed to reject almost anything.
Exactly. Currently, we try to physically filter their blood or use toxic therapies to wipe out their immune cells. But imagine an engineered exosome specifically designed to seek out and bind to those pre-existing alloantibodies, neutralizing them in circulation.
Oh, wow.
Or, better yet, a vesicle carrying apoptotic signals designed to selectively target and destroy the specific memory B cells producing those antibodies.
So we would be moving from merely trying to avoid triggering an immune alarm to deploying microscopic agents that actively dismantle the alarms that have been ringing for years. We could take a highly sensitized, untransplantable patient and effectively clear their immunological memory, turning them back into a blank slate for a new organ. It completely changes the paradigm from managing rejection to actively erasing it.
It really is the ultimate promise of nanomedicine.
Well, thank you for joining us on this deep dive into the microscopic battleground of your own immune system. To everyone listening, keep questioning the consensus, and we'll see you next time.